Okay, welcome to Cellular Respiration Review. Purpose is for you to hear this one more time before the test, or as many times as you want before the test, because you can access this through YouTube. So, Cellular Respiration Review, the key here is that you see the pattern. Don't focus necessarily on the numbers, even though I might throw some numbers out at you. The idea is that you see the patterns from one area to the next. So, this first image gives you a nice overview of the three main pieces of cellular respiration: glycolysis, Krebs cycle, and the electron transport chain. I will highlight some of the different ways that ATP is made and where these locations are, especially at the very last slide that I go through. So, first part, glycolysis. We're going to do this in the cytoplasm, where the cytosol, as this diagram is saying. We are going to make some ATP, but the key is that we're going to be shuttling some things, hopefully, all the way down to the electron transport chain, and we're going to be shuttling some things into the Krebs cycle. So let's take a look at the next piece, which is the Krebs or glycolysis. So let's look at this real quick in detail. So with glycolysis, the key here, as the word kind of says, glycolysis, right? So glycolysis. We're talking about glyco, sugar, lysis, to break or weaken. So we're talking about breaking sugar. So the first thing that comes in is glucose, right? We have pretty much a 10-step process where the first 5 we consider the investments, because we're actually going to put in some energy. And then the last 5 we call the energy output. So the reason we call it the investment is we're going to put ATP in. So you can see ATP is getting put into the molecules, and here's the molecules moving down. Anywhere where you see an arrow, there's an enzyme involved, except it's not in this diagram to keep things a little bit simpler. So glucose turns to glucose 6-phosphate, and ATP is added in. So anytime that you see ATP being used or formed, there's going to be an enzyme involved. So an enzyme here, you should already kind of know, that is a kinase enzyme. So whether or not we're using ATP or down here making ATP, we're going to be using that kinase enzyme. Then we get to the output, and the reason it's the output is not only do we make ATP, you can see here, but we also make NADH, which is key because NADH is going to be our electron shuttle. NAD plus turns into NADH, so we're literally taking electrons out of glycerol 3-phosphate, or G3P. So G3P is something that we also saw in the Calvin cycle of photosynthesis. G3P is a product of one turn of the Calvin cycle. So this is a molecule that's seen between both of them. It's a nice builder molecule. But when we take electrons out of G3P, the electrons go into NAD+, making it NADH. So electrons into this molecule, so NAD+ gets reduced because it gained electrons, and G3P got oxidized. Remember that redox, right? Red, ox, right? It's a combo, so one molecule will get reduced and another molecule will get oxidized. So it's an exchange of electrons. The enzyme that facilitates this, which you do need to know, are called dehydrogenase. I'm going to run out of room. ACE enzymes, right? So they're the ones that help allow this to happen at body temperature. So what we ended up doing, we used 2 ATP here. We ended up making 4 ATP and 2 NADHs, and then what we're left with, carbon chain-wise, is 2 pyruvate. So we start with glucose, which is a 6 carbon, so 6 little spheres. So each little circle is going to represent a carbon. And eventually we end up with pyruvate, which is 3 little circles. So we really did not change the carbon chain length, which we'll pick up as a pattern that we'll be looking at as well. If that chain decreases in size, right, it goes from 3 to 2, then we have to lose carbon somehow. And the way that we lose carbon in cellular respiration is through CO2 production. All right, next. All right, the Krebs cycle. So now our pyruvic acid, or pyruvate, is going to be translocated into the mitochondria. So all that back here So we're just, you silly, why they put those so close. Sorry about that. All this is happening out in the cytoplasm. If pyruvate is in the presence of oxygen, the pyruvate will be translocated into the mitochondria. But if it's not, we'll talk about near the end here, we're going to have to ferment these molecules in one of two ways, depending on what type of organism you are. So the Krebs cycle, what they're not showing on this image is that pyruvate has kind of a transition, right? So a little extra spur where it feeds in to turn into acetyl-CoA, which then will fuse into this cycle here. So there's a little piece before this, a spur before this, so to speak. So that little spur will actually make some CO2. So right now you should already have picked up the pattern, and we're going to make some NADH. Okay, so the NADH that gets made, we're going to shuttle on forward. The CO2 can be exhaled, and pyruvate went from 3 carbons down to acetyl-CoA, 2 carbons. So in the chain right now, oxaloacetic acid, as you see it right here, is actually 4 carbons long, and we're going to bind in 2 carbons, so isocitrate is 6 carbons long. Okay, and then anytime CO2 is released, the carbon chain shrinks, so you should know that 6 carbons, alpha-ketogluteric acid, fancy name, is 5 carbons long. Notice that we made some NADH here, made some NADH here, made some NADH here, make some NADH here. So there's a lot of NADH being made in the Krebs cycle. We also make FADH2, which is very similar to NADH, and that is an electron shuttle. So we're going to use those later on in the electron transport chain. Notice as well that we make 1 ATP for 1 turn of the cycle. So 1 pyruvate coming in, we basically get 8 NADHs. We get 2 FADH2s, and we get 2 ATP, right? So that is for a turn of the Krebs cycle, right? So we're going through this through 1, 2, 3, 4. Actually, this is 2 turns, sorry. So this would be 2 turns. So this would be 2 pyruvate will yield this. My fault. Trying to multitask. This is not working for me. So 2 pyruvate will yield 8 NADHs, 2 FADH2s, and 2 ATP. Why 2 pyruvate? Because remember, glucose went through glycolysis, producing 2 pyruvates. So keep that in mind when you're talking totals here. Since we're making ATP right here, it's going to be a kinase molecule again. We'll talk about that. It's called substrate level phosphorylation, making that ATP. And these would be dehydrogenase enzymes, right? Dehydrogenase enzymes. All right, so Krebs cycle is done. So you think about it, from our original carbon chain, we really don't have any carbon left because all the CO2, as you can see, gets released in the Krebs cycle. So all the CO2 is released, all of the NADH and FADH2 is made here. Very little ATP so far, though. So the original carbon chain is pretty much gone, but all the energy from that original carbon chain is pretty much now in shuttles, NADH and FADH2, right? So the Krebs cycle, main big things, releasing a lot of CO2, making a lot of NADH, and making a lot of FADH2. All right, Krebs cycle goes to the electron transport chain now. So locations-wise, we did glycolysis out here, right? We did the Krebs cycle in here in the matrix, and now we're doing the electron transport chain between the matrix and the inner membrane space along this membrane right here, which is called the inner mitochondrial membrane, or the cristae. So, electron transport chain, we have integral proteins inside the membrane. We have a difference now between one area and the next. We can actually set up what's called a proton gradient. And the way that we do that is we're going to do active transport. So these proteins, 1, 2, 3, will serve as pumps. They will pump H pluses, which we consider as protons, into the inner membrane space, so the orange area, from the yellow area. So we're going to pump H pluses from the matrix into the inner membrane space. How does it get the power to run active transport? In past, we've said powering pumps requires ATP, but when in sense, it just needs some sort of energy. The energy that we're going to use is NADH and FADH2, which you can't see on this diagram here. So NADH will drop off its electron that it picked up, and it turns back into NAD+, which is key because we're going to recycle these molecules. That electron then excites this protein and allows the pump to happen. It passes the electron, hence the chain, right? And it continues downward. Every single time it works, it loses energy. So by the time it gets to the third protein, there's very little energy left in that electron, and oxygen is your final electron acceptor. When oxygen takes that low energy electron, it also incorporates some H pluses, and it turns into water. So now water has a very low energy electron, which is going to be key because if you're thinking circle of life, that low energy electron then is going to be given to chlorophyll A in photosynthesis for the light reactions, right? Non-cyclical photophosphorylation. So now all these electrons, right, passing through NADHs, right, pass. We got all these H pluses pumped up into this area. So now this area is super saturated with H pluses. They're all stuck there. They're charged. They can't just willy-nilly go through the membrane. They have to go through some sort of facilitated diffusion doorway. And that facilitated doorway is ATP synthase. When it comes back downhill through ATP synthase, we get some conformational changes, and ADP turns into ATP. The fact that we have this coupling of the electron transport chain and ATP synthase is chemiosmosis, which we see in plants as well, generating lots of ATP. And since our original source of energy was the oxidation of sugar, right, the loss of electrons from a sugar, we call this manufacturing of massive quantities of ATP oxidative phosphorylation. Okay, so that is the electron transport chain. So everything that we did is pretty much all done, right? The only thing we have to address now is what if oxygen's not present? So if oxygen's not present, we're not going to get those pyruvic acids, or the pyruvate here, into the mitochondria. So we have to do something else. Now, the big reason that we do any sort of anaerobic reaction is not to make more ATP, it's actually to make NADH, so, or NAD plus from NADH. So remember, from our glycolysis, we made ATP. That's great and dandy, we need that. But we also made NADH. NADH has to drop off its electrons. Where does it normally drop off its electrons? It sends it all the way into the mitochondria, and then it comes back as NAD+. Well, if we don't have oxygen, we cannot get into the mitochondria. So fermenting is the way of dropping off electrons. In both of these systems right here, lactic acid and alcoholic, the NAD+ is used into pyruvic acid to turn back into, NADH is used in pyruvic acid to turn back into NAD+, so we can do another round of glycolysis and get some more ATP. The difference is lactic acid is still 3 carbons long, whereas alcohol, ethanol, is 2 carbons long. So in ethanolic fermentation, we release CO2. So goal, fermentation, recycle your NAD+, and we're not really making ATP. Okay, so that's fermentation. So let's look at the final results, and we'll wrap up this review. So glycolysis input, we're obviously putting in glucose. We're going to put in NAD+, and we're putting in ADP. Outputs, the glucose turns into pyruvate. The NAD+ turns into NADH, and the ADP turns into ATP. Now, since we did have 5 in and 5 out, you could technically play that an input was ATP and an output was also ADP. But I'm just talking about nets here, so it's pretty much the net amount. We're putting in more ADP than we are putting in more ATP, right? Enzyme C, we see the kinase enzyme here forming ATP, and we see dehydrogenase. I write too fast. And then the ATP production, we're doing substrate level phosphorylation. all those fancy words you remember that you went home and told your parents about to show them that you're learning so much. Input, Krebs. We're going to take the pyruvate. We're going to send it to the Krebs cycle. Right? We're also going to put in some more NAD+. We're also going to put in FAD, right? And the pyruvate turns into CO2, and the NAD+ turns into NADH, and FAD turns into FADH2. Remember, we also get some ADP turning into ATP. So we're getting ATP here, we're getting ATP here, we get a whole lot of ATP over here in the electron transport chain. Enzymes seen, once again, same ones, kinase and dehydrogenase. And the only way we made ATP here is, once again, substrate level phosphorylation. Okay? Remember, the goal is we're making lots of these things, CO2, NADH, and FADH2. Then for electron transport chain, we're going to take the NADH and the FADH2, send these over to electron transport chain. The other input that we're going to put in is oxygen. Oxygen turns into water, right? Output, we get lots of ATP. We also get NAD+ and FAD, right? So these, NAD+ and FAD, they can go back over here to the Krebs cycle or back over here to the glycolysis, get refilled, and recycled. Enzyme seen, it is a dehydrogenase, although we did not focus on it much. The key one that you need to know, enzyme seen here in the electron transport chain, is ATP synthase, at least for our purposes. If you go onward, you'll realize that all the proteins that are found in the cristae actually have names, and they're pretty much enzymes as well. ATP production, we call this oxidative phosphorylation. Okay, and that's it. If you have questions, let me know. Otherwise, I wish you the very best of luck. I know you'll do well. I know you don't maybe have a lot of confidence in yourself at this point, but realize it's almost over. So hang in there. You'll do great. Talk to you later.